Hello everyone. Welcome to our channel Biochemistry for Student. Here we are going to learn some important topics related to biochemistry that cover all the aspects started from cell to nutritional component to the genetic or the molecular biology. Here we are going to learn about recombinant DNA technology today. We learn this recombinant DNA technology in two parts, part 1 and part 2. In part 1, we will go through the important biological tools, or what we can say components that required to go through this recombinant DNA technology process. And the second part, we will go through the recombinant DNA technology process. So as we know, it is a very important technology in today's era. For example, if we say in today's era, so many medicines in the form of protein or hormone we are taking, that is produced with the help of this recombinant DNA technology. For example, insulin, that is a most commonly used hormone used in the treatment with the patient of diabetes mellitus, particularly type 1 diabetes mellitus, is produced with the help of recombinant DNA technology. So it is a very important technique to learn and to understand. So we continue with the recombinant DNA technology. In this, we will first go through the restriction endonuclease enzyme and vectors, and we also learn about the complementary DNA in this part 1. And in part 2, we will go through the procedure of recombinant DNA technology and the applications of this technology. So this recombinant DNA technology is one of the recent advances in biotechnology. Though it is an older one, developed in 1973 by the Herbert Boyer and Stanley Cohen, but still we are saying that it's a recent advance because it has a very much potential to do so many newer things with the help of this recombinant DNA technology. So what it is actually recombinant DNA technology means? The simple recombinant DNA technology is technology which allows DNA to be produced by artificial means. So we can produce DNA artificially. This procedure has been used to change DNA in living organism and may have even more practical uses in the future also. Okay, so it is an area of medical science, as I told you, though it is almost 3 or 4 decade older technology, but still we are saying that it's just a beginning to be researched in concerted efforts. Genetic recombinations, if we see this word, that is just literally exchange of information between two DNA segments, that is genetic recombination, and this is usually occurs within the same species. The genetic material exchange in the two different segments of DNA within the same species, that is a very common occurrence. But by artificial means, when a gene where artificial means when a gene of one species is transferred to another living organism, that is our recombinant DNA technology. So that is this is recombinant DNA technology when we transfer a gene of one species to another living organism, that is recombinant DNA technology. So this technology starts with the isolation of gene of interest, that is called target gene. So what gene we want to get or we want to amplify artificially, that is called target gene. Then the target gene is inserted into A plasmid or a phase, that is a kind of transporter, or what we can say carrier, that is known as vector, to form a replicon. So this replicon, once produced, replicon is nothing else but a vector having our target gene. It will be introduced into the host cells. After that, it either that gene undergoes the expression, it produces the protein, or it may be remain as such. So we get the copies of that particular DNA. The clone replicon is referred as a recombinant DNA. What this replicon what we get, that is called recombinant DNA, and the process is known as recombinant DNA technology. Fine. So for this process, as I told you in this part, what we are going to learn is what are the tools required for this genetic manipulation or the genetic amplification. That is enzymes. That includes restriction endonuclease enzyme, very important, polymerases, exonucleases, endonucleases, DNA ligases, and etc. Apart from that, the important thing what we required is target gene we can produce by producing the complementary DNA, and the last one is vector that we can say as a carrier we required to transfer this complementary DNA to the host cell. They are plasmid, bacteriophages, cosmids, and etc. So we start with this enzyme, the most important one, that is restriction endonuclease enzyme. That is known as a molecular scissor. This restriction endonuclease enzyme is known as a molecular scissor. Why it is known as molecular scissor? Because it functions like scissor and cut the DNA. It cut the DNA at very particular site, or you can say restricted site. That's why it is known as restriction endonuclease. This enzyme acts as a palindromic site. This enzyme acts as a palindromic site. Palindromic site means a site that have a same, if we read it from the front or back, it is the same meaning, like race car. If you read from the front forward, that is RACE car, or in the backward it's a race car. The same way in the double stranded DNA, the same sequence is found in the opposite order. Here you can say GAATTC, on the other strand GAATTC. So that is a palindromic site. So this restriction endonuclease enzyme act at particular site, that is restriction site having palindromic sequences. So these enzymes, they are mainly of two types. One that produces the sticky end at the DNA, and second one is that produces the blunt end. We see how. So you can see this is double stranded DNA having a palindromic sequence, or we can say restriction site. At that site, this restriction endonuclease act particularly at site and produces the stretch that having a single strand on both the side. When this cut DNA molecules having a sticky end attach with the same sequence on the other side, it will get stick to the opposite strand. So this is the restriction endonuclease that produces a sticky end. You can see this is the two different DNA acted upon by the same restriction endonuclease enzyme. It produces the same sticky patches, and here you can see there is a change. There is a change of that fragments. Okay, so that bind with each other at the sticky end. The second is that enzyme that produces the blunt end, and that act on the middle, of this DNA and produces the two blunt ends of the DNA, and that can combine with the other DNA, and that can be joined with the help of DNA ligases. Okay, so this is restriction endonuclease. This restriction endonuclease enzyme has been isolated from the bacteria, and based on the bacterial strain from which they have isolated, the name of this restriction endonuclease is given. At present, there are more than 900 or we can say thousands of restriction endonuclease have been identified from different 250 bacterial species. Now, for example, here you can see ECO-RO, that is the most commonly used restriction endonuclease enzyme. The ECO means that is derived from the Escherichia coli strain, that is R, and the first enzyme of this strain. So that is ECO-R1. So hence the name is given likewise. For example, if we take this one, HIND3, that is Haemophilus influenzae D strain, and in that third enzyme. So likewise the name are given to the restriction endonuclease enzymes. And based on their activity or the characteristics, they produces either sticky ends or the blunt end, and that helps in the recombinant DNA technology. Now, the second part, what we require is target gene, or what we can say a gene of interest which we want to amplify or which we want to express to produce the protein. So isolation of specific gene from human DNA is a laborious process. We can get this target gene from the gene library also, and if it is not available, then we can produce by producing the complementary DNA. So why it is important to get the DNA? We see, for example, if we want a beta globin gene, beta globin, that is a beta chain in the hemoglobin structure, that is around 1.6 kilobase per in length and represents only 0.00005% of total human gene. So you can understand how difficult it is. It's like a searching of needle in a haystack. Only a single gene that represents only 0.00005% of total human genomes. To overcome this problem, we have what we can say a technique for the production, that is preparation of complementary DNA. So we can produce complementary copy of DNA to overcome the problem of getting isolation of this gene. We'll see how. The complementary copy of DNA, it's a simpler process compared to identify particular gene from the cell. So we have to start with the messenger RNA, because when we talk about any protein that is selectively expressed because of selective or selectively produced in particular cell because of selective expression of that particular gene. For example, we take insulin. Gene of insulin is present in each and every cells of body, but it has expression, selective expression, we can say in the beta cells of islets of Langerhans. So this insulin is produced in the beta cells of islets of Langerhans. So gene for that insulin is expressed only in the beta cells. So from that gene, the first mRNA is synthesized, that is messenger RNA, with the process of transcription, and this mRNA undergoes the translation process and it produces the insulin. So the only beta cells have messenger RNA that translate for the insulin. So we start with that messenger RNA. From the specific messenger RNA, we will produce this DNA, that is complementary DNA, by the reverse transcription process with the help of reverse transcriptase enzyme. We'll see the steps how we will synthesize or we get the messenger RNA and how it convert it into complementary copy of DNA. So first we isolate all the RNAs of that particular cell, that includes messenger RNA, rRNA. After that, separating or isolating that RNAs, we will allow this RNAs to pass through a column that contains oligo-DT, oligonucleotide having thymidine. So when this messenger RNA or RNA pass through that column, the messenger RNA will get attached with this oligo-DT, because during post-transcriptional modification, messenger RNA will get poly A tail, and this poly A bind with the oligo-DT. So it will be retained in that column. After that, we will elute out the messenger RNA with the help of chemicals from that column. And again, this beta cells of islets of Langerhans not only contain the insulin as a protein, it has other proteins also. So there will be different messengers RNA. Out of that different size coding, we will get particular messenger RNA for insulin. Now the specific mRNA identified after passing through the column, and this messenger RNA will be now acted upon by reverse transcriptase enzyme for the synthesis of DNA. And that time we will add deoxynucleotide triphosphate with that will polymerize to form a double-stranded DNA. So we will get RNA DNA hybrid. What we will get is RNA DNA hybrid. So we have started with the messenger RNA, and at the end what we get is RNA DNA hybrid with the reverse transcriptase enzyme. And this process is known as the reverse transcription, and we get a complementary copy of DNA. Again, the same thing. We have isolated a messenger RNA with the poly A tail, and that will be acted upon by the reverse transcriptase enzyme in the presence of deoxynucleotide triphosphate, and we get double-stranded copy of DNA, that is complementary DNA. Now, as I told you in the beginning, in this process, once we get the target gene, we have to introduce this target gene into the host cell. And for that, what is required is called carrier, and that carrier is known as vector. So in order to introduce the human gene, that is what we can say a target gene, into a bacteria or other host cells, we require a carrier that is known as vector. And in that, there are different varieties of vectors available. We are going to discuss about plasmid, bacteriophage, and the cosmid. They have their own properties, and based on that they are utilized in the recombinant DNA technology. First, plasmid. It is the most commonly used vector for the process of recombinant DNA technology. It's just a simple, small, circular duplex of DNA molecules present in the bacteria that usually confers the antibiotic resistance to the host cells. We know the bacteria get or they becomes resistant to the particular antibiotic, and this antibiotic resistance they confer by this plasmid. The important thing about the plasmid is they replicate independently of bacterial cell division, and that can be also easily separated from the host bacteria. So these are the important characteristics. Because of that, plasmid is an ideal vector for the recombinant DNA technology. But the only drawback of plasmid using as a vector is it can accept only short stretch of DNA having 6 to 10 kilobase per long. That is the only drawback of the plasmid. The second vector used in recombinant DNA technology process is a bacteriophage. It is having a linear DNA in which foreign DNA can be inserted at several sites using restriction endonuclease enzyme. And the advantage of using bacteriophage as a vector is we can introduce a slightly larger size of DNA fragment that is around 10 to 20 kilobase per long. The third vector used for the recombinant DNA technology is cosmid. They are nothing else but the plasmids having cos site, that is DNA sequence required for packaging of lambda DNA into the, that is a viral DNA into the bacteriophage. The advantage of using cosmid as a vector in recombinant DNA technology is it can accept very large DNA fragment that is around 35 to 50 kilobase per long. So depending upon the gene of interest, we can use or we can choose the vector. So this is all about the important biological tools required for recombinant DNA technology. I hope all of you understand the initial what we required for the process of recombinant DNA technology in part 1. In part 2, we will learn about the process of recombinant DNA technology and we'll see the various applications of this technology. For more updates, just you can visit our Insta account, that is at the biochemistry for student. I hope all of you will get the idea behind this technology. Please like, share and subscribe our channel, that is biochemistry for student. Thank you.